Hi there! Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world.
Hi, everyone, and welcome to From Lab to Launch by Qualio. I'm Meg, your host. Thanks for tuning in today. Before we jump in, we'd love it if you would rate the podcast and share it with any of your science nerd friends. We know you have some. If you'd like to be on the show, please fill out the application linked in the show notes. We're grateful for all the interest we've had lately. So today we have Dr. Jack Hoppin, CEO of Ratio Therapeutics, and Dr. John Babich, Chief Science Officer of Ratio Therapeutics. The team at Ratio Therapeutics is developing a suite of innovative technologies to develop best-in-class targeted radiotherapeutics for the treatment of cancers. They recently just opened a 19,000 square foot headquarters and R&D facility in Boston to continue their exciting innovation. We'll dive into how Ratio started, their growth, and what you can learn from their journey from lab to launch. All right, let's bring these gentlemen in. Welcome to From Lab to Launch. We're glad you're here.
Thanks for having us. Wonderful to be here.
Yeah. So Jack and John, to kick things off, we have a wide range of listeners on this show, some chemistry nerds, others like myself, not so much. Can you help us better understand what Ratio Therapeutics does as if you were explaining it to your grandmother?
Sure. Go ahead. I have to learn another language as my grandmother, but the simple basis for targeted radiotherapeutics, right? So what we're doing is administering a radioactive drug that localizes in cancer, and just like typical radiation oncology, is delivering energy into that cancer in a way that will kill the cancer. The difference really is that where typical radiation oncology uses machinery and directs beams that, from multiple different angles so that, you can basically hit the tumor in a crisscross pattern without hitting lots of other tissue at the same intensity. We have to administer a drug intravenously. And so our objective really is to design a drug in such a way that the most of the radioactivity that gets administered to a patient will localize in that tumor or in that cancer and stay there for a prolonged period of time so it can actually release its energy, deposit its energy into the tumor to affect the kill. So it's a field of nuclear medicine. This is the use of radioactive materials for diagnosis and therapy. We're particularly interested in the therapeutic applications. We have a series of approaches to that we're working on right now at Ratio.
That sounds really cutting edge. Is there a particular cancer that you're focused on, or is this applied to kind of all cancers across the board?
So, every time we contemplate a cancer, we have to match the cancer type with a particular target that cancer might possess, and the target being a particular protein that sits on the surface of the cancer so that we can get access to it from the blood supply. And so we have to think those things are not, they don't just sort of fall out of trees, so to speak. And so we have to really think about that. Right now we have a program that's ongoing in prostate cancer with a particular target. And we have a program that's a bit more broadly focused, and that is to look at really a component of a tumor. So if you think about the difference between a cancer and a tumor is really a swelling. The lump somebody would feel in the breast, or the lump you would feel in a prostate digital exam, or some lump that you would find that you think maybe this could be a cancer. That lump is generically in Latin referred to as a tumor, but it may be a cancer, it may not be. So what it turns out is that there are a lot of cancers that accumulate tissue side by side with the cancer cells that are from the host. And so it's giving out lots of signals. It wants to grow. It wants nutrients. It wants blood supply. And in the process of actually sending out signals, it'll recruit something called fibroblasts. And these are the kind of cells that would make a scar form if you've got to cut yourself. The healing part of your body that actually gets recruited into sort of stitch or cut, form a scab, and then basically renew that tissue. So you get the cancer cell recruiting these various what are known as fibroblasts, because of the signaling, they get turned on. When they get turned on, they present a fibroblast associated protein, which is known as fibroblast activating protein alpha, or FAP. And this turns out to be something that's expressed on many types of cancers in the combined environment that the cancer cells and the host cells make. So they call it the tumor stroma. This is a sort of mixture of cells from the host and the cancer itself. And so we're going after that. It's called FAP. We have a diagnostic program with one of our pharma collaborators, Lantheus, on the development of a diagnostic to image a variety of cancer, and also image any type of disease that provokes a fibrotic response. We also have the rights for the application in therapy, and this would have application along many different cancers, from pancreatic to breast to colorectal to lung, and also, say, liver cancer. There's a variety of cancers we can apply that to, and that's what we're developing right now.
That's very exciting. I have known people personally affected by each of those three cancers you mentioned. So amazing work that you guys are doing over there at Ratio Therapeutics. Kind of shifting gears a little bit, you've both founded companies in the past, but why did you start Ratio together, and how did you two get connected?
Well, we've known each other for a long time, but had never worked together. Yeah. And you may want to tell you tell a story. Yeah, and I do. Oh, so
it's like asking a couple how they met. I love it.
So, we met a long time ago, but in 2018, at the time, John was a professor at Cornell in Manhattan, and at the time, I was overseeing a company I founded, in vitro, that was really kind of the leaders in imaging drugs and discovery, and also as an endpoint in clinical trials. And many people, this class of drug that John just nicely described is really an emerging, not a new concept. Radioactive iodine was FDA approved in 1951, but it's in the last decade that it's really gotten a big movement. Novartis and Bayer have joined the space, and Novartis has recently gotten a big prostate cancer drug approved. And so we were seeing lots of drugs in this class and over the last 5 or 7 years. And it's really, to John's point about where the drug goes, how it accumulates, and does it have the right ratio of resident time in the tumor as compared to any excretory or off-target resident time. And John showed up with something he had invented and developed and really thought through. He's been a pioneer in this space and studied a lot of drugs. And my core competency is really in the studying of the pharmacokinetics of these drugs and trying to model them and understand where they go and quantify. It's a very quantitative sport, as we always radio label at first and take a PET scan or a SPECT scan. We do a scan. We actually can see where and when and where over time the drug is. And so John showed up with this concept, and I immediately said, that is the right idea. That is brilliant. It's really about tuning the pharmacokinetics of the drug. And John invented a platform where the binding moiety is targeting cancer itself. He mentioned two targets, PSMA and prostate, and FAP, the pan cancer target. And then another part of the molecule at a much lower binding affinity, or worse binding affinity, it kind of hitchhikes albumin, which is a ubiquitous protein in your blood. Your coffee mug full of albumin flowing through it at all times. And this extends the bioavailability of the drug in the blood. And then that really allows more of it to get into the tumor and often changes its clearance mechanism. And so right away, I was excited by this. And that was a previous effort, and that drug was sold to Bayer in 2021, which simultaneously spawned Ratio. Don quit his day job. I had subsequently retired, according to my LinkedIn page, briefly. And we came back and started the company with a partnership with Bayer, a partnership with Lantheus that was already discussed, and raised some capital and built the team. And now we sit in this headquarters. We're 35 people, very technical team, very interdisciplinary sport. You need chemistry, radiochemistry, biology, this quantitative pharmacology. You need physics, you need radiation dosimetry. It's just a lot of PhDs and not that PhDs, a lot of scientists of varying backgrounds here, a very technical squad, and it's going well.
Great.
And I'll just say, one of the reasons I reached out to Jack is I saw that this technology would be amenable to actually the application of modeling. So this is a really tremendous skill set that Jack has and his team at his previous company, and that we've expanded on here as well. And to understand, can you actually mathematically model what's happening in a human, and then take that modeling information and then, through trial and error, make the molecule match what you expect the model to do. And so this is kind of, we talk about math meeting medicine. This is kind of where this comes together. The idea that, there is so much to do when it comes to quantitatively assessing where these molecules go, because they have an impact on therapeutic benefit and on the side effect profile. And there's a lot of calculation on this, right? So it's not just pharmacology for pharmacology's sake. It's also pharmacokinetics, and the delivery is important, and the ratios between how much gets delivered to the tumor versus other tissues is critical. So this is a real nice opportunity to use all this kind of expertise to sort of design drugs and understand drugs in a way that we maybe haven't understood them before.
That's great. It sounds like a great partnership between yourselves, Jack and John. Speaking of partnerships, we've mentioned a few big names in the industry like Bayer and Merck, and some others. How are you going about commercializing and executing on your go-to-market strategy with these big partners?
Yeah, I think there is a really large appetite in this market for novel drugs. And I think what's great about this field is the interdisciplinary nature. I think there are many people who felt like, I have a failed drug, I'm going to repurpose it, put radioactivity on it, and make a targeted radiotherapeutic. Or there's people who say, I have a new isotope, and I'm going to find a binder, and I'm going to have a new drug. But it truly takes knowledge both in pharmacology, in standard pharmaceutical sciences, which is obviously very challenging given the failure rates, and in nuclear medicine. I think that's what we wanted to build here. And so it's great to have partnerships, because you expand sort of your skill set and domain knowledge. We're still a very small company of 35 folk. And so to have partnerships where you can bring something to the table, but also, with the might of some of these larger enterprises, not just from a dollar perspective, but also from a domain knowledge perspective. So I think we're very eager to partner with folks, and that's working out.
That's great to hear.
Yeah.
One of the things we like to discuss here on our podcast is quality management, since Qualio is a quality management software. Can you tell us a little bit about the quality culture you're seeking at Ratio? It seems like you're definitely after product quality, but what does that quality culture look like, and how do you balance quality and product development?
I'm gonna let John answer this, because he is truly a foremost expert in putting. We both have a lot of experience putting radioactive drugs in people, but I will jokingly say, like, there's actually no stress, no pressure putting radioactive drugs into people, right? There's no quality. No, sorry, go ahead, John. Yeah, so the bar is. I think we take quality. We take it very seriously. Not to imply others don't, but the issue with quality really starts at the very, very beginning of everything we do, right? So, and this is another significant, I would say, attribute of Ratio, in that we're trying to make sure that we dial in the quality from the first time we make a molecule to when we track the molecule, to how we curate data about a molecule. And I'm talking about chemistry, obviously, at the very beginning, but also on, let's say, protein-based assays, cell-based assays, preliminary evaluation in animals, ultimately preliminary evaluation in humans, making sure that quality is built into data generation, data archiving, and then data, at the point where you actually have to look at data, how do you curate the data, and how do you present the data So I think quality in this organization comes at go. And so there's also, the QA functions of, CMC and the QA associated with clinical trials. I mean, that to me is a given, but it's almost like, when you start to do that, you have to have quality. You couldn't do it without it technically, right? You would be going against GMP and all the required obligations one would have from a regulatory perspective. So I think quality assurance really starts much earlier for us in our tracking of information about the molecules that we generate and how we move that information in order to make decisions, in order to move drugs forward. So I think I think it's almost part of our DNA now that the quality of how we acquire information, how we store it, and how we evaluate it is kind of in. I'm trying to think of another analogy, but it really is baked into our systems. And then, of course, there's, I would say, typical in a way of quality assurance relative to clinical trial, site selection, vendor selection, the quality assurance packages that go along with CMC. One of the things about this space, which is kind of different than maybe many other spaces, that whatever you're going to do with therapy, you actually have to make the drug. And so it's unlike some drugs that can be made in large quantity and then stored in a tablet form or even in a solution form and put in a fridge or put on a shelf. Because it's radioactive, it's like making ice cream in the summertime, right? It's you make it and it's disappearing as soon as you finish making it. And so you have to actually make the drug over and over and over again. So say for a clinical trial of 30 patients, you might have to make the drug 30 times for 30 patients. You might wind up with a batch per patient only because of the way scheduling occurs. So there's a lot of quality assurance that's built into the manufacturing, and it has to be, at a level that is, I would say repetitive is the best way I can say it. And then, of course, there's always the quality assurance that gets implemented in the readout and the regulatory filing. So it really is in our DNA here, and it's in a big part of how we make decisions is based on the quality that we put into those decisions.
Yeah, I will just add, and John nicely stated it to John's point of the ice cream. In addition to being ice cream, it's really a micro dose of ice cream. So the mass doses we deal with, when we use for imaging, we use tens of micrograms, and for therapy, it's low hundreds of micrograms. And if you just think about a 200 milligram Advil and taking a thousandth of that, it's just a really, sometimes things don't work. And this is one of those strange fields where some people do a run and they don't get what they want. So a lot of effort has to be go into making sure that you're able to deliver drug on that day, unlike having, to John's point, a stockpile or lyophilized or whatever. So I guess, the gene therapy folks on the call would maybe say ours is tougher. And I don't think we'll disagree.
Yeah, no, that's a point.
That's another thing. It just, it just presents with challenges and there are failures.
Yeah. I love that when the margin of error is so small, quality in your DNA really helps from go. That's great.
And I think, to Jack's point, we have to go through a quality control every time we release a batch. So that has to be live and on the fly, right? It's not like you can wait and people are on the phone, people are sending emails, people are releasing, remotely, right? Once the whole data comes in, you can evaluate things. So it's I wouldn't say it's touch and go, but it's certainly high pressure on the day of the manufacturing.
Certainly sounds like it. Speaking of high pressure, maybe you recently had raised for $40 million a few years ago. Congrats, by the way. How did you approach funding, as often is the number one business problem for founders in this life sciences industry?
We had a very good founding, in the sense we had a couple of strategic partnerships out the gate, and were able to just raise some seed capital, and then kind of based on previous experiences, had business contacts that were enthusiastic about a partnership, the team we built, continue to build. And so we were able to raise capital through really more angel, high net worth family offices and existing kind of partnerships we had. That didn't mean it wasn't incredibly stressful. It just means that it was it was different. That's the approach we took. And right in our Series B, we are exploring more institutional, more classic venture capital institutional money, but there's a lot of partnerships out there, so it'll probably be some high.
Great. Well, switching gears back to yourselves, if you could go back to the start of your career and give yourself one piece of advice, kind of knowing how you've gotten to where you are now, what would it be?
I don't know. We got here. Yeah.
Yeah.
I didn't get here. I give myself different advice.
What advice would you give yourself to get here easier, maybe?
Oh, easier?
That I would have a hard time thinking there was an easy way to get here. I would say the joke I tell people when previously I started a few companies and ran one for a decade that did very well. The joke I say to people if they're starting a company is like, when they say, what would you do differently? I say, probably not do that. It's really hard. It's a lot of work. But then again, there's no real, there's no credit without risk of blame, and there's no success without risk of failure. So I guess, I don't know. I think maybe eat better. That's probably Get more sleep.
Sleep better.
Getting sleep.
A little more health.
Yeah, I think, I think the other, so I've been involved in a previous company we took public, and I think one of the things that's really critical is to understand your investor base. I think that's really, I would say I've had now 3 experiences, and that is probably one of the most important things to understand is who's investing, why are they investing, what are their expectations, communication is key. I'm not saying we're not doing that. I'm just saying that to me, that seems to be a crucial part for particularly companies in this space where revenue is not right around the corner, right? So you have to understand this sort of time frame of your investor, what their expectations are, and then make sure there's lots of communication, because bad things happen, right? If it was easy, there'd be a drug company on every corner, right? So. But the rewards would probably be a lot less. But when you do have success, so I think it's to understand that component of the business, because it's a big part of the business. It's not insignificant, and I think also to make sure communication is foremost upfront with your investor base.
Yeah, I think that's great advice.
Yeah.
Yeah.
A good IP. I think the other thing is getting a good IP attorney, intellectual property attorney is critical. Again, when you're going into a business like this, you really need to have a technological position, and you have to make sure you're protected on that. So, I mean, these aren't things we haven't done. There are things we have done, but I think they're crucial to getting established on the right footing, and then making sure you go in the right direction, because things happen, as we know.
Yeah, I think that's great advice for any investors or innovators looking to join the space. Our last question that we like to wrap up here is with a fun one. For each of you, if we ran into at a bookstore like Barnes & Noble, in which section would we find you in?
I could say, for better or worse, I would like to read like history of science.
History of science?
Yeah, I'm happy to read any history book, but I typically would be reading some sort of history of the science, math or physics, typically. But that's usually what I read.
Very interesting.
I would not be in that island necessarily. I would
history and math is definitely not its own island. It's not its own island.
No, I would probably be in the nonfiction section. Yeah. And I just, not that I don't have math books, but I think it takes me a while to get into fiction, and it just doesn't take me as long to get into nonfiction. So I tend to follow those books up more. So but I like to understand, other people's journeys in these sort of situations, and also the psychology. The psychology behind decision making is really fascinating to me, right? Some of Malcolm Gladwell's books, Blink, the Tversky's books, and Canahan. I mean, it is just so many ways to understand how you think and how you react to information, how the world reacts to information. It's always humbling to have people study that and to see how kind of goofy we are at the end of the day, and when we think we're so driven by detail, and then a bird goes by and we make a change in decision. You're like, okay, that's a very solid decision to make. So maybe I shouldn't pay attention to birds so much. But there's a lot more to, and this goes. I'll bring the quality into it, right? It's like, how do you actually figure out the best way to make a decision? How's the data? What is the data really telling you, and that you're not fooled by it, or you're not fooled by your own biases, right? So that to me is kind of where I find my head in the book.
Well, I hope you have lots of summer reading ahead. Thank you so much for joining us from Lab to Launch here. I learned so much about Ratio Therapeutics and what you guys are up to. I will be following along. Where can our listeners go to follow along and connect with you?
Well, certainly our website. Yeah. And
thanks to the great group at Russo Partners, our LinkedIn page is really active. I admit I don't have a big social media presence personally. We're certainly on social media, and LinkedIn.
Yeah.
Right. Well, we'll add your website and LinkedIn in our show notes. Thank you so much for joining us today, Jack and John.
Thanks,
awesome meeting you, Meg.
Nice to meet you. Good luck.
Likewise. Thank you.
Here.
Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time.